I'm here today to talk about ChemAxon's technology in Reaxys. As you can see, a lot of names on the slide. Originally, the presentation was thought to talk only about the excerption part of Reaxys, but then we decided to put a little bit more into this presentation. So all of these people, or most of these people, have added some slides and some comments, which I hope will be able to bring to you as good as possible. Allow me some quick words to myself. So my role at Elsevier is the product owner of the excerption software. We call it Intuitive Excerption Interface. So basically, it's coordinating requirements between business, content, and developers to make sure that the taxonomies which we are using are up to date and that everything is coordinated between the taxonomy team and the other stakeholders. And yeah, in the background, of course, we are always looking for innovative ways to improve the excerption, both in terms of quality and efficiency. And also automatic methods and machine learning plays more and more a role in the day-to-day work. In my speech, at first, I would like to give a very, very quick introduction into Reaxys. I noticed that some of you are not so familiar with Reaxys, so I think this is really a good idea. Then the workflow of Reaxys is mentioned, followed by the manual excerption in a little bit more detail, the architecture of Reaxys, and then structure queries and some future projects, and everything with the aspect of the ChemAxon tools which we are using there. So this is quite a lot, as you can see. At first, a very short introduction into Reaxys. So chemistry, as you all know, plays a crucial part in our day-to-day life. Think only pharma products, chemicals like in your car, in your iPod, wherever. There are so many companies and A&G people who are doing research in chemistry, so this is really, really a big market, as you know. In essence, chemistry deals with two core questions. First, which compound has the property I want to have, and how can I make this compound? These two questions has been our passion for quite some time. So actually, everything started already in the 19th century with the first edition of the Gmelin Handbook, which is about inorganic chemistry. Still in the 19th century, the first edition of the Beilstein Handbook was published. Then last century, in 1989, Beilstein and Gmelin for the first time go online. In 1992, Crossfire was launched. Perhaps some of you still know about Crossfire. In 2009, then Reaxys was launched, which was followed then by a complete overhaul of the interface and everything, which led finally 2016, so 1.5 years ago, to the new Reaxys interface, and now also medicinal chemistry RMC is part of the platform. So the core of our philosophy, Reaxys aims to deliver immediate access to information, and not only access to information, but more and more also analytical aspects and analytical ways to investigate the data you have found. The new Reaxys interface is now quite clean when you come to the first screen. So you just have the field which allows you natural language questions or drawing a structure immediately. But of course, you can enhance it to a very detailed search mask like you know it from the previous versions of Reaxys and Crossfire very previously. The text which has been entered by the user in natural language is transformed into search queries, and the search queries can be edited, and after the first search, some suggestions are given if not 100%. The answer is not 100% clear. Some search suggestions or result suggestions are given to the user. And he can use this one then as a starting point to make his search strategy more detailed. This is just to illustrate the amount or the different facts which we have in Reaxys. So the facts which we have in Reaxys are displayed in several ways. This is just to give you an impression how many facts we have. Of course, there's filtering and sorting, so you can really go down to the core of the problem and reduce your set to some 10 compounds, compounds or reactions. Okay, so what does Reaxys offer? A lot of compounds. Currently, we have 140 million substances, more than 55 million records for bibliographic data, more than 45 million reactions with 500 million properties, and finally, now completed also by bioactivity data and target data. Okay, this was Reaxys in a rush. Now some words to the workflow. The way how the data get into the database starts at an electronic version nowadays, no more paper files, but only electronic version, which at first is automatically extracted. The output of the automatic extraction is then sent to the manual excerption. As you can imagine, the automatic excerption is by far not perfect. So we manage to excerpt some compounds, some reactions, if especially in patents the text is well given and has a good format. But for the big part of the data, we still need a manual excerption. So the excerptors who we have, they excerpt structures, reactions, and of course the many facts which we have. And for the excerption, one of the core things of our work is Marvin sketch. I will go into the manual excerption in a little bit more detail in the next slides. Once the excerption file is finished, it is sent to the Reaxys database, so it's no direct editing on the database, but a file is prepared, which is then sent to the database and registered into the Reaxys database. Okay, now a little bit more to the manual excerption. This is the excerption software, which I have mentioned earlier. The intuitive excerption interface. On the left side, you can see the original source, which is either a PDF document or an XML document, which is displayed in an HTML viewer. In the middle pane, you see the objects which have been excerpted. In this case, you can see compounds, but you see here some more tabs, so also reactions, facts, assays, targets, and some other things which are relevant for pharmacological data can be displayed and excerpted. But today, it's mainly about the core of our excerption, the structures and the reactions. So here on the right side, you can see a so-called excerption mask. In this case, it's the identification mask of a compound. So you can see the center of it is the structure. The molecular formula is given automatically. If there's a label for the compound, it can be given here, the names, synonyms, and even some modifications and other data. As I said before, Marvin sketch is used for entering the structures. What we did in cooperation with ChemAxon was, for example, that ChemAxon tailor-made some of the lists which are behind the Marvin sketch for our special needs. For example, the two-dimensional stereo descriptors like here absolute, racemate, or steric, which I think is quite unique in this way for Reaxys. So they implemented this for us so that we can use it as it is has been done in Reaxys and Beilstein previously. This is just to show that we also excerpt Marcus structures. The excerption of Marcus structures is also based on Marvin Sketch as a structure editor. So the scaffold of the Marcus structures are entered with Marvin Sketch using the options, the various options which we have for R groups or for aliases, which can be entered here, so that the way how the Marcus structure is given in the article can be shown more or less identically in the Reaxys database. The next thing which we use in the manual excerption is the enumeration tool. The enumeration tool is something which we developed in close cooperation with ChemAxon. On the left side, you can see here is a table in a patent, and very often it occurs that a scaffold is given together with such a table with these R groups, and if these are, for example, prophetic compounds, the excerptors actually need to enter all these compounds during the excerption process. In order to accelerate this, we developed the enumeration tool together with ChemAxon. So in this tool, the user just has to draw the scaffold. This he has to do. But then he is able to add these substituents, which are given here in this table, just in a separate window. And after he or she has done it, he can simply combine those ones which are relevant for the excerption, and then automatically create compounds from this process. So here it is shown how some of these listed substituents are drawn here. The attachment point is added now, and pressing OK, you will see that all these appearing here now. Some can be added, and for the other substituents, the same can be done. And then in a very quick selection process, the user just can put them all to this table here, press export, and then all these compounds, all these combinations will be created in the excerption software in one go. You will see this now in the navigation pane. If I scroll down here, all these compounds which are a little bit gray here, these are all these compounds which have been created right now. There are some more features, like for example the label, which is added automatically. So for the excerptor, this helps making the work much more efficient than before. And I would like to emphasize that the cooperation here for building this tool with ChemAxon was excellent. So there was always a very good support from especially Efi and Arpad. And yeah, I would like to thank you for this. It's really great. Next, very quickly about the Reaxys architecture. The Reaxys architecture, if you remember the slide which I have shown you earlier of the workflow, on the right side here we have the Reaxys database. The Reaxys database is part of a search engine cluster, which is depicted here. So one part of this search engine cluster is Reaxys. The whole architecture is embedded in the Amazon workspace. The user who is, in terms of ChemAxon tools, working with Marvin.js in order to add structures. This is the front end, and the user works with ChemAxon here with the Marvin.js tool. In the background, the search requests which are coming from the user are sent to the Reaxys backend. There we have the tool JChem Web Services, which is needed because Marvin.js would be too slow if it comes to more complex calculations. Once the search request has been sent to the business layer sends the answer back to the Reaxys backend. And here again we have a tool from ChemAxon, which is Mole Printer, and Mole Printer then makes the images for compounds and for reactions and displays them finally in the Reaxys user interface. So the structure queries which we which a user draws in the Reaxys user interface, as I mentioned before, is supported by ChemAxon's Marvin.js again. The key Reaxys use case, as many of you know, is reaction searching and synthesis planning. And it's necessary very often to draw very complex reaction queries and compound queries. And this is enabled by Marvin.js. In this case, you can see a reaction search query, and some of the features which Marvin.js is offering are given here. For example, the R group logic is given, the atom-atom mapping, position variation bonds, diverse R groups, atom lists, and you name it. This then is the search result. This, for example, is one of these images which has been prepared by a mobile printer, as I mentioned before. And here you can see some experimental data and some other facts which belong to this reaction. Again here, I would like to mention the good cooperation with ChemAxon in the first release of Reaxys, so which I mention here as the old version. This one was not yet fully adapted to the Reaxys user. So we get some complaints, and yeah, this was discussed with the ChemAxon guys, and they quickly came up with a solution. So in the new version, which I think was released last week or will be released this week, we have several requests from customers we have implemented now. So for example, the Reaxys generic groups are much better visible now. We have atom-atom mapping included. We have the maximum substitution count included, which was a big request. And also updated the templates, which were very basic before and now are a little bit more sophisticated. Yeah, again, very good cooperation. So, I'm approaching the end of my speech. I would like to mention some ideas for the future, which we have. We think about predictive tools. We would like to use Reaxys data and Reaxys Medicinal Chemistry data in order to build predictive tools. Two examples I would like to give you. Think, for example, of bioisosteric replacement. This is important when you want to enhance affinity, if you want to correct an ADME problem, or perhaps sometimes it may be necessary to overcome an IP issue. So for these problems, it would be good to know what would be the result of such a bioisosteric replacement. Another example, target prediction. Target prediction still may be called the holy grail of the medicinal chemist. The primary activity, favorable or unfavorable side effects, or perhaps even drug repurposing. And also here we are very confident that we will have some points of cooperation with ChemAxon. For example, Marvin.js again will be part of such things, or ChemAxon server tools, of course, in order to transform the molecules into smiles or 3D structures. Yeah, this is basically the end of my speech. Thank you very much for your attention. If you have any question now, I'm open to answer them.
You could see from this example how flexible Marvin Sketch and Marvin.js usage can be if somebody understands our API. And which also Ralph mentioned that they get user feedback, and they get back to us and ask how together we could develop something which more suitable and intuitive for the user. So I really thank you for that cooperation and the way how we did it.
You're welcome.